So I've mentioned stomach function a little bit in our broad overview, but I wanted to go into more detail on the functions of the stomach. So there are three that we want to know. The very first one I've talked about. The number one and three we've talked about. Number one was protein digestion, and that is through production of HCl and pepsinogen by parietal cells and chief cells respectively. Actually, I'm going to skip ahead and I'm going to show you the picture. So parietal cells are in blue. They will secrete HCl, and chief cells here, also in the body of the stomach, will secrete pepsinogen. Now, HCl denatures proteins. That's what it does, and that helps protein digestion. And it also converts pepsinogen, which is inactive, to its active form, which is pepsin. And pepsin will cleave peptides. So that's it for protein digestion. Our second function is intrinsic factor secretion. And this is again from the parietal cells in the blue. An intrinsic factor, the function of it is to bind vitamin B12 and bring it to the terminal ileum to be absorbed. If you do not have intrinsic factor, vitamin B12 does not reach the terminal ileum. So what will happen is that you will have low vitamin B12, and vitamin B12 is needed to make, basically for red blood cell development. So if you don't have it, you're going to get an anemia. It's going to be a megaloblastic anemia. That's a key word that we'll talk about in the heme lectures. Finally, the stomach is a reservoir for ingested food. Remember why do we want this again? Do you remember from our previous lecture? Remember that digestion in the small intestine is a very slow process. You don't want to overload the digestive system, so you need to modulate it, slow it down, and you need to store that food somewhere that you just ate. So, to illustrate the importance of this, sometimes we'll remove the whole stomach. It's called total gastrectomy. That's the surgery we'll do. And what's going to happen, you're going to have, if you eat too much, you're going to have a bunch of hyperosmolar food boluses directly entering this small intestine because there's no more reservoir. And all that hyperosmolar food is going to draw in liquid and water from the intestinal cells into the lumen. And so with all that water, you're going to get diarrhea, and it's going to be something called dumping syndrome. It's colicky, abdominal pain, and diarrhea. So that's no fun. So we've talked about three different substances that are secreted so far. We've talked about HCl, talked about intrinsic factor, talked about pepsinogen. The next one to talk about is gastrin, which I've already told you about. And gastrin is made in the G cells of the antrum of the stomach. Remember what gastrin does? Remember that was a regulatory substance that helps in digestion, and it helps in digestion through indirectly increasing HCl release, and it's going to also increase GI motility. And the very last one to know about is mucus. The mucus cells release mucus. And why do we need mucus in the stomach? It's because you need to protect this whole stomach lining here from all that corrosive HCl that you're releasing. HCl is great for breaking down proteins, but you don't want to break down your own stomach. So you need to line the stomach with mucus to protect it. So that's it for our stomach function and the stomach, different substances released in the stomach. Next, we're going to focus on gastric acid secretion. Now, there are three different phases that will lead to release of gastric acid secretion, will lead to regulation of gastric acid secretion. We've talked about this first phase already a little bit. This is the cephalic phase. Remember, cephalic for brain, example, encephalitis. So this is basically when you're looking at food, when you're thinking about it, when you smell it, when you taste it a little bit, and you send all these nice signals to your brain, and your brain is going to start that digestive process. It's going to prime your body to get ready for that food. So it's going to get some cholinergic and vagal mechanisms to stimulate gastric acid secretion. So next is after you finish smelling and looking at the food, you finish taking your pictures on Instagram, and then you eat it, and the food hits your stomach, and it's going to cause stomach distension and chemical stimulus, and that's going to be the gastric phase of gastric acid secretion. And so these stimuli, which we've already talked about in our substance videos, will lead to gastrin release, and gastrin will stimulate histamine, which will then lead to gastric acid secretion. So gastrin indirectly leads to gastric acid secretion through histamine. Our very last phase is called the intestinal phase. So that's when the protein-containing food enters your duodenum. And what's going to happen here? Remember, we already talked about this. Remember, do we like acid in the duodenum or not? Remember, that is not good. So what we're going to do is that we're going to have control mechanisms that reduce gastric acid secretion so that our enzymes work better. So those are our three phases of gastric acid secretion: cephalic, gastric, and intestinal. And now let's talk about substances that actually stimulate gastric acid secretion. So what substances do we actually directly act to lead to gastric acid secretion? There's three of them. We already talked about most of them. So number one was histamine, which we talked about, which directly stimulates those parietal cells. Number two is acetylcholine. Remember, that's the vagal. It's from the vagal stimuli, and that also directly stimulates parietal cells. And finally, which was the one that was indirect that I just talked about? The indirect one was gastrin, remember, and that it increases histamine release, and then obviously histamine, so gastrin increases histamine release, and histamine will stimulate parietal cells. So these are our three, and I just remember them because there's Hag, so it's a sourdough Hag that cause gastric acid secretion from parietal cells. Okay, so next is pancreatic secretions. So the pancreas secretes many digestive enzymes, very important for digestion. Pancreas will secrete trypsinogen. Remember, inogen basically means it's inactive. And it is converted to its active form, which is trypsin, by enterokinase. Entero is basically means the gut, the small intestine. So, enterokinase is an enzyme on the brush border of the duodenum and jejunum, and it converts trypsinogen to its active form. And trypsin, is important because it activates other pro-enzymes from the pancreas. So all the enzymes the pancreas release are not active yet. That's because if it releases and it's active, it's going to just eat up your whole pancreas, and you don't want that. Okay? So it needs to be secreted in its inactive form and then eventually activated in the right place. So the thing that it does is the pancreas helps digest all your nutrients, all the different ones: carbohydrates, proteins, lipids. And it's going to do that through the release of amylase, proteases, and lipases respectively. So amylase for carbohydrates, proteases for proteins, and lipases for lipids. Finally, pancreatic juices. Pancreatic juice is an isotonic solution. Has many different ions, and the ones we want to take note of are bicarb and chloride, because these are the two that are going to actually vary with the flow rate. So it's actually kind of weird that this is a little graph here of the pancreatic juice release. So this is flow. This is higher flow here. Higher flow. At low flow rates, you have high chloride levels. So this is chloride. At low flow rates, you have high flow levels. High flow levels of chloride. But that's going to decrease as your flow increases. So your levels of chloride decrease as your flow increases. Bicarb is the very opposite. Bicarb at low flow rates on the left side is very low. But when your flow rate increases, so does your bicarb. So that's the difference. So very last part is nutrient absorption throughout the GI tract. And this is basically a synthesis of everything we talked about. So, carbohydrates. Will there be any digestion of carbohydrates in saliva in the mouth? Yes, there's amylase. Is there any digestion of carbohydrates in the stomach? Remember we talked about stomach function. Was there anything about carbohydrates? No, because stomach doesn't do anything to the carbohydrates. Next is the pancreas. Is there anything that the pancreas does? Yes, remember the pancreas does pretty much digest all of the different nutrients, so there's more amylase in the pancreas. And the intestine will add a couple other various enzymes to help finish carbohydrate digestion. So next is proteins. Anything in the saliva? No, saliva doesn't do anything for proteins. What about the stomach? Remember the stomach does have that protein digestive function, and so it's through pepsin and hydrochloric acid, so HCl as well. That's annoying. I'm sorry. Excuse me. And then next is the pancreas. What does the pancreas do? Remember, the pancreas secretes proteases. Remember, they're secreted as inactive proenzymes, and then those are activated by trypsin. What about the intestine? Intestine also has proteases. So finally, lipids. Let's talk about lipid digestion. Any lipid digestion in the saliva? Yes, there's actually something called lingual lipase. So lipase can digest lipids in the stomach, in the mouth, so lingual. Stomach, any lipid digestion, no. Finally, pancreas, any lipid digestion, yes. Pancreas digests all of them, so it secretes lipases, and there's no lipid digestion in the intestine.